We have already done double fertilization. I believe everything related with double fertilization is very much clear to everyone in the class. Let's talk about it once again. This is the female gametophyte and antipodal. This is egg cell, right? This one is egg cell, which is surrounded by, surrounded doesn't mean you have so many synergids. You have just two synergid. We have learned in female gametophyte, no, 8 nuclei is formed, and in the center you have secondary polar nuclei. It can be polar nuclei or secondary nucleus, you can say. Basically, it's a diploid structure. You have 2n over here. Now, what is going to happen? Pollination happens, which leads to germination of pollen tube, and this pollen tube goes in and leaves into the embryo sac two male gametes, right? The first male gamete would fuses with the egg cell. So egg cell will fuse with the first male gamete, forming the zygote. Right? This zygote is going to be 2n. One egg cell is n, and the male gamete here is again n, that gives you 2n, right? So the zygote that's formed is 2n. And the second male gamete, it will fuse with the secondary nucleus, or you call it as the diploid region, or we can say the secondary nucleus. You must have learned as polar nuclei also, so don't confuse yourself, polar nuclei and secondary nucleus. तो जो secondary nucleus है उसके साथ जो second male gamete है वो fuse करेगा. Then how many n would be there? 1, 2 for this and 1n for this. Total how much would be? 3n. So it's a triploid structure which is going to be formed. So now you have to remember जो center में structure बना है ये है हमारा endosperm. What is that? Endosperm. What is endosperm? What is the function of this endosperm? We are going to understand that also. We have already learned the 3 types of endosperm: nuclear, cellular, halobial, right? So this was the double fertilization, which involved 2 events. One is syngamy and triple fusion. I hope it is very clear to everyone. Once fertilization happens, what all things would happen? So you have to remember it like this: post fertilization changes that's happening. So first post fertilization changes जो हमने पढ़ लिया है, endosperm development, जो कि 3 टाइप के थे, right? The nuclear, cellular and halobial type. Now the second event that happened after double fertilization is embryo development. Now there is a question, ऐसा क्यों होता है embryo development endosperm के बाद develop होता है? ऐसा क्यों नहीं पहले embryo development और फिर endosperm development? क्योंकि fusion तो पहले हुआ था egg cell के साथ, तो कायदे से the zygote should develop into embryo first, no? But it's not like that. Endosperm को पहले develop होने इसलिए देते हैं क्योंकि endosperm देता है nourishment to this growing zygote, right? The zygote is going to develop, it has to get nutrition. तो कौन देगा nutrition? It will be given by the endosperm, right? We learned 3 types of endosperm: nuclear, cellular, halobial. The function of endosperm is to provide nutrition to the growing embryo inside the floral gynoecium part. That's the female gametophyte, right? We are talking about the ovule. So now let's understand development of embryo. So we'll all be on page number 11. So the heading is 1.11, page number 11, development of embryo. So we are on page number 12. Page 12. But on page 12 figure 1.15 seems to be little absurd, right? एक्चुअल में पूरा एम्ब्रियो सैक भी इसके अंदर बना हुआ है, इसलिए आपको अलग सा लग रहा है वो डायग्राम. एक्चुअल में इतना सिंपल डायग्राम है, इट्स नॉट डिफिकल्ट एट ऑल, इट्स वेरी सिंपल. So let's see how the things are done over here. How the embryo is developing. तो ये जो डायग्राम है आपके सामने, it is the dicot embryo. Dicot embryo. Dicot means which has got two cotyledons. जब आप मटर को ओपन करते हो, when you open the pea, और जब ऊपर का स्किन निकालते हो तो अंदर आपको दो cotyledons दिखते हैं, तो हम उसे बोलते हैं dicotyledons, right? बट अगर आप राइस को खोलते हो, राइस के हस्क को रिमूव करते हो, कैन यू गेट टू कॉटलिडन्स? नो, ओनली वन. सो दैट इज मोनोकॉट, राइट? सो व्हेन देयर आर टू कॉटलिडन्स, the embryo development diagram would be different. So let's understand how the development happens. So सबसे पहले क्या हुआ? Male gametes has fused with the egg cell, right? And once the male gamete fuses, जो antipodals हैं और जो synergids हैं वो सब disintegrate हो जाते हैं, they just vanish. Okay, all what is left is this structure, that is the zygote, right? It's 2n diploid structure. Okay, अब ये 2n में multiplication होता है, there is formation of टू सेल स्ट्रक्चर. जो आपके सामने है ये टू सेल स्ट्रक्चर है, जिसको हम बोलते हैं प्रो एम्ब्रियो. क्या बोलते हैं हम इसे? प्रो एम्ब्रियो. सो यू कैन ड्रॉ दिस डायग्राम, दैट विल बी सफिशिएंट फॉर यू. नाउ इन दिस प्रो एम्ब्रियो, टू सेल, आउट ऑफ दिस टू सेल यू कैन सी द डिफरेंस. दोनों का डिविजन बराबर बराबर नहीं हुआ, इट्स नॉट इक्वल डिस्ट्रीब्यूशन. एक सेल छोटा है, दूसरा सेल बड़ा है, राइट? जो छोटा वाला सेल है, इट इज़ ऑन द टिप रीजन एंड इट्स ऑलवेज टूवर्ड्स द चलाजल रीजन. दैट्स द चलाजल रीजन, चलाजा. सो आपने जो एनाट्रोफिस ओव्यूल में चलाजा एंड माइक्रोपाइल का डायग्राम का पोजिशनिंग समझ लिया होगा. सो माइक्रोपाइल इज डाउन, दिस इज द माइक्रोपाइलर रीजन. ओके, सो now when you look at this diagram, let's say, wait a minute, I have to draw it once again for you people. This was the embryo sac and this was the zygote. Remaining all has gone. We are left with endosperm. Endosperm develop होता है, endosperm grow करता है. Okay, अब ये जो zygote है, this should reach towards the food. This is providing food, no? जो endosperm है वो food देता है, तो it should reach towards the food. So how will it reach towards the food? There will be this, जो basal cell है ना, वो divide करने लग जाता है and that part is called as suspensor region. देखो लंबा, long, elongated structure बनता है. I'm just removing it. Elongated structure बनता है. Can you see this? Why is there so much of elongation? There is elongation just because ये जो embryo है, it has to get nourishment. तो nourishment के लिए इसको push करता है towards the endosperm. Inside there is endosperm, no? तो towards endosperm, this suspensor cells are, suspensor cell means the bottom cell, right? The zygote was divided into two, pro-embryo stage. And from that pro-embryo stage, the filamentous suspensor cells, somewhere around 6 to 10 cells are formed, and the suspensor cell will move and it will actually push the embryo towards the endosperm. That's why it goes high. It goes tall and tall. Now let's see the condition of embryo. अब embryo का shape देखते हैं हम. ये shape कैसा है? It's circular, right? Embryo is circular. This is all endosperm region, जो कि इसको nourishment दे रहा है, right? After circular, next shape, there will be division and the shape will be the shape of the heart. Very popular among you people. So first was globular structure, then heart shaped, And the next structure is this horseshoe shaped. एग्जाम में कई बार ये आया भी था. What is the sequence in which the dicot embryo develop? What is the shape? Sequential shape. Okay, so you have to remember the first will be the globular structure, round structure that multiplies, and then it would be the heart shape, and then it would be the horseshoe shaped. अब आप लोग बोलोगे ये ऐसे ट्विस्ट क्यों हो जाता है? Because there is not much space in the endospermal region, that inside the female gametophyte it doesn't have too much space. Okay, so it bends down and at the end forming this extra growth that's happening here, that forms the cotyledons. What is that form? Cotyledons. So you have to learn this diagram. One cell stage, zygote, फिर it divides into two unequal parts. One would be the larger and another would be the smaller. The larger is called the suspensor initial cell, जो कि embryo को push करता है towards the towards the food, right? Endosperm. And the smaller cell that we have, it's the embryonal cell, embryo, right? That develops. अब सस्पेंसर के अंदर का जो डिवीजन है उसको हम बोलते हैं ट्रांसवर्स डिवीजन. देयर इज ट्रांसवर्स डिवीजन जिसके वजह से 6 to 10 सेल्स बनने लग जाते हैं. एंड सबसे लोएस्ट पार्ट, मतलब जो इसको बोलते हैं लोएस्ट रीजन, we call that as hypophysis. What is the function of this suspensor cell? It is to help the embryo to get nourishment from the endosperm. For a minute, let's go there. So please underline here. The suspensor helps in pushing the embryo in the endosperm. Okay. So पहला structure है ये वाला A structure. दूसरा structure है pro embryo stage. Okay. B. देन प्रो एम्ब्रियो में जो सस्पेंसर है वो 2 सेल बनाते हैं, S1 and S2. एंड हियर एम्ब्रियो ऑल्सो डेवलप्स. दे मल्टीप्लाई लाइक 2, 4, 8 लाइक दैट. दे मल्टीप्लाई and they keep forming different, it's like complete division happening over here in the embryonal cell. अब यहाँ पे okay, so I think it's easy for you to draw first diagram, second diagram, third diagram and the fourth diagram, right? अब demarcation भी आप लोगों को आनी चाहिए. E means the embryo, H means ये वाला जो सेल जो एम्ब्रियो को टच कर रहा है सस्पेंसर का सबसे लोएस्ट सेल, उसको बोलते हैं हाइपोफाइसिस. देन वी हैव सस्पेंसर सेल जो नीचे पूरा है. अब इसी स्ट्रक्चर को हमने एंडोस्पर्म के साथ यहाँ पे दिखाया है, राइट? दिस इज शोइंग the antipodal tissues here, and this is embryo, hypophysis, then suspensor cell. और ये जो डायग्राम है ये देखिए आप, can you tell me what kind of endosperm is found here in this dicot diagram? I'm going to stop it right over here. What kind of endosperm formation is found in this diagram? D diagram में कौन सा एंडोस्पर्म फॉर्मेशन है? आई वॉन्ट द आंसर, देन वील कंटिन्यू विद दिस.